this is how the structure of skeletal muscle will be. The outermost, you have the muscle belly, and inside, if you do a cross section, each muscle belly will contain the outer covering, muscle fascicle, and inside the fascicle you have multi-unit of muscle fibers, and inside the muscle fibers you have the myofibrils, and then it contains myofilaments, which includes what? What are the myofilaments? Actin, myosin, thick and thin filaments. So this is the structure. Now we will go with the sarcomere. Sarcomere is the single contractile unit of the muscle, as like a cell, which is the functional unit of living organisms. The sarcomere is the functional unit, contractile unit of the muscles. Okay, and two important components in the sarcomere, you have myofibrils, actin and myosin, and the sarcotubular system. So this sarcotubular system, this is the sarcotubular system, the tubular system which is present in the sarcomere. Okay, you have two types of tubules, T tubules and the L tubules. As the name T tubules, you have T-like invagination of the sarcomere, the sarcolemmal membrane. So here you have the ECF. This T tubules is making the ECF to go and communicate with the interior of the sarcomere also. Okay, and you have, this is the L tubules, L-shaped tubules. Okay, this is called as the L tubules. And at specific point, you can see the junction of one L tubule, another L tubule, and the T tubule. So this is called as a triad, where the L tubule and the T tubule connects together. Okay, clear? So mainly the T tubule acts as a signal transmission. Usually the extracellular fluid is the one which is going to bring the stimulus. So that stimulus, because of the invagination of the T tubules, it will also go and reach the interior of the sarcolemmal membrane. And then L tubule, this L tubule contains the cisternae. This part is called as the cisternae, where the calcium stores is present. Why you need the calcium? For the action potential to take place. Okay, so once when the signal is reaching the extracellular fluid, that will come and reach the membrane of the sarcomere. Since the membrane is having the T-shaped invagination, the potential, the signal will also reach the T tubules entire length, and this T tubules will give signal to the L tubules, and this L tubules, which is having the calcium stores, it releases the calcium. And what are all these structures? They are the myofilaments, actin and myosin. This calcium will go and reach the myofilaments, and these myofilaments will get activated, and it will be involved in the action of muscle contraction. Okay, and this is about the sarcotubular system. And now coming to the sarcomere. Okay, so all these T tubules, L tubules you will see over here. Okay, and now this is one striation, this is another striation in the skeletal muscle gross. And the space between these two, this is called as sarcomere. Okay, so the line which is dividing one sarcomere with the another sarcomere, you will term it as a Z line. Okay, so the space between two Z line is called as the sarcomere. Okay, and inside in between the Z line, you have thick filaments and the thin filaments. Okay, so this is what is called as the thick filaments. This entire one which is in the center is the thick filaments. Thick filaments is made up of myosin. Okay, so thick filament is made up of myosin. And these structures which is remaining attached to the Z line, they are called as thin filaments. Thin filaments is made up of actin, troponin and tropomyosin. Okay, thick filament is made up of only myosin, but the thin filament is made up of three structures: actin, troponin and tropomyosin. Okay, so first you just see this concept is very important, and if you don't understand, it is quite confusing. The questions will be asked with this, mainly based upon what is Z line, what is M line, H zone, A band, I band, all these things. Okay, so remember first what is sarcomere? Space between two Z line, that is called as sarcomere. Z line is the like a cell membrane, the space between the two cell membranes. When you see in the gross, you have the striations like so. This is one Z line, this is another Z line. In between this, that is what you are seeing here as a sarcomere. Okay, clear? And in the center of the two Z lines, you have the thick filament. So this is what is the thick filament. And this entire thick filament you call as the entire thick filament you call it as the A band, which contains only thick filaments. Okay, so this is the A band, and this contains thick filaments. And now next you see with the thin filaments, this is the thin filament. The thin filaments are attached to the Z line. Okay, so this one is called as the, and the space, what is this demarcation? Here also you have the thin filaments, but here it is having the alternation of thick filament, thin filament, thick filament, thin filament. But this partition, which is containing only the thin filaments, and on the other side, this is called as the I band. This I band, it is also called as isotrophic. This is A band anisotrophic. Okay, clear? And then next, in the center, where you have only the thick filaments. A band is having alternative thick and thin filaments, but it includes the entire length of the thick filament, that is A band anisotrophic, in the sense it is not same, because it is having the alternative of thick and thin filaments. But in the center, which is having only the thick filaments, that is called as H zone. And the centermost part between two Z lines, it is called as M line. Okay, M line, it is just the centermost between two Z lines. There is no specification like thick filament or thin filament. That is the centermost part of a single sarcomere. Okay, clear? So this is what is the arrangement of the myofilaments in the sarcomere. And now we are going to compare this. What happens if the muscle is going to contract? If a muscle is going to contract, what will happen? That is what we are going to see. Okay. So what will happen if the muscle is going to contract? Clear with these arrangements first? What is Z line? The space between the two sarcomeres. that is Z line. The space between two Z line, you call it as a sarcomere. And what is A band? That is the entire length of the thick filaments. What is I band? Which contains only the thin filaments. What is H zone? Center of the A band, which has only the thick filaments, no thin filaments. M line is the central demarcation between the two Z lines. Okay. And now we will see the sarcotubular system in the muscle contraction. See, this is the T tubules. This is the T tubules invagination in the sarcomere. And this is where you will have the L tubule, where the calcium store is present. Okay. In the presence of a stimulus, here you have to consider two important receptors. One is dihydropyrimidine receptor and the ryanodine receptors. These two receptors are involved in the muscle contraction to initiate the signal in the sarcotubular system. Okay, so this dihydropyrimidine receptor, it is present in the T tubules, and the ryanodine receptor, it is present in the L tubules. So once when the signal is reaching the sarcolemmal membrane, this signal will travel to the invaginated segment of the sarcolemmal membrane also, that is the T tubules. And then this dihydropyrimidine receptor will make the signal to transmit, and then it is reaching the L tubules. This L tubules releases the calcium. And this calcium, whatever is released from the calcium stored in the store of the L tubule, it will go and bind with the ryanodine receptor. So now this calcium is the basis for muscle contraction to take place. So calcium is the most important electrolyte which is required for muscle contraction. And what is the another function which we have seen for the calcium? In the neuromuscular junction, release of neurotransmitter, calcium plays a major role. Okay, so now what will happen once when the calcium is released? This is the three-dimensional view of the myofilaments: actin, myosin, troponin, tropomyosin. Okay, so this is the thick filament, which is having the thick filament. Which band? Thick filament, A band, which is having only the thin filaments, I band. What is H zone? The center of the thick filament, that is the A band, which contains only the myosin. Okay, so this is the structure of the thick filament. So single unit, if you want to see, you have a single myosin like this, head and the tail. Okay, so this head will have ability to contract like this. See, for example, this is the myosin. Okay, this is the body portion and this is the head portion. So the myosin head will bend, it will move like this, it will come back like this. Okay, and now we will see first about the thin filaments, then I will explain you the muscle contraction, molecular mechanism, how it is taking place. So thin filaments contain, what are all the thin filaments? Actin, troponin and tropomyosin. So this green colored bead-like structures, they are the actin. Okay, and in between that you have a thread like this orange colored structures. They are the tropomyosin. And then, so this actin is like a bead-like structure, and then tropomyosin, and you have in between troponin. So in the actin, you have a site called as myosin binding site. Specifically in the actin, you have a myosin binding site. That is the site where the myosin is going to bind with the actin in the presence of a signal during muscle contraction. If there is no signal, the actin and myosin will not remain attached. Okay, so this myosin binding site where it is present in the actin will be always covered, closed in the absence of a signal by the structure called as tropomyosin. Okay, so thin filaments contains actin, troponin, tropomyosin. Actin is a globular structure, the bead-like structure which is arranged together in a single thread-like manner. Okay, so this actin is containing a myosin binding site where the myosin is going to go and attach with the actin in the presence of a stimulus. If there is no stimulus, this myosin binding site of the actin will be covered by the tropomyosin, which is also one of a thin filament. Okay, this tropomyosin is like a thread-like structure. Okay, so actin will be like this, twisted, like a beads, twisted together, and in between the actin you have a, here and there you have the myosin binding sites. Those myosin binding sites, if you twist a rope along with the beads, like that you will have the tropomyosin. What this rope of tropomyosin is going to do, for example, consider my nail as an actin, and that nail is containing the myosin binding site. Okay, and this is the tropomyosin. During twisting process, the tropomyosin is covering my nail because that is the myosin binding site. So it is covering in the absence of a signal, stimulus. Okay, now there is a presence of stimulus. The muscle is going to contract. So what will happen? My nail is exposed. Still tropomyosin is twisted, but it is just moving away its location to expose the myosin binding site in the actin. Okay, clear? So this will happen when in the presence of stimulus. Which chemical is released? Which electrolyte is released from the sarcotubular system? Calcium. The tropomyosin will expose the actin to go and but attach with the myosin because in the presence of calcium. If there is no calcium, it will not expose. It will always cover it. Okay. And where this calcium is coming and giving signal to the tropomyosin is to the troponin, which is also a troponin is also an thin filament. Thin filament has actin, troponin, tropomyosin. So this troponin, this is the troponin. This troponin has three sites. Okay, like I, T, C. So troponin is like this. This troponin has three sites. One is I site of troponin, troponin I, T site of troponin, C site of troponin. This I site, T site, C. Okay. So this T is the place where the tropomyosin will bind. C is the place where the calcium will bind. I is the place where the actin will bind. Okay. In the tropomyosin is like a rope. In between the rope, I am forming some knots. Like that, the troponin will be present. It is also twisted with the actin, the bead-like structure of the actin. So once, when the sarcotubular system releasing the calcium, that calcium is coming and binding in the C site of the troponin. The calcium will come and bind. This calcium is giving signal to the T site of the troponin. What signal? Now there is a signal, the muscle has to contract. So you move your location to expose the myosin binding site in the actin. It will move. And now the site is exposed for the myosin to come and bind. Okay. So as like you have seen, actin is containing the myosin binding site. In the myosin also you have a actin binding site. Okay, understand? Actin is having myosin binding site. Myosin is having actin binding site. Okay, so now this is the thin filament which is having twisted rope-like structure. Imagine the tropomyosin and it has exposed. Because imagine the tropomyosin has exposed the actin, actin containing the myosin binding site. Now this is the myosin. Okay, this is the myosin, head of the myosin. This structure is what my finger is going to explain now. Okay, the single myosin. Okay, and this nail, consider this nail, the myosin head containing the actin binding site. Okay, clear? And now consider the calcium has made its function, the troponin binding with the calcium, making the tropomyosin to expose the myosin binding site. Now the myosin is ready to go and attach with the thin filament, actin. Okay, for this process, this is the myosin, it is bending, making it to attach. You need energy in the form of ATP. Okay, and then next, it has attached. Now the muscle has to contract. So contraction, the head should move. So the head will move in the presence of ATP. After that, it will detach. Again, it will go and bind with the another myosin binding site of the thin filament. For example, consider this is the thin filament. Okay, the same myosin binds, attaches, detach, again bind, attaches, and then detach, again bind, attaches. This will happen from both the sides of the Z line. Okay, so both the sides, the myosin going and binding, attaching, 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 makes the Z line to come closer. Okay, so both the Z line comes closer, because in the Z line, see this picture now, both the sides you have the Z line, and both the sides this is going and binding, attaching, 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 make it to come closer. Okay, so the both Z lines comes closer, and now we are going to see the arrangements which has shortened, which has remained in the same position after the muscle contraction. So what will be the condition of both the Z lines after muscle contraction? Is it in the same length or it has shortened? Shortened. Good. And then what is the position of A band after muscle contraction? The length of A band after muscle contraction. Both the Z lines comes closer. See, now consider this is the Z line and this is another Z line after the muscle contraction. What is the position of A band? It remains same because the Z line is attached only with the thin filaments. A band is having the thick filament which remains same. See now, this is the thin filament, this is the thick filament. The thick filament is remaining in this position only. It is binding, attaching, it is binding, attaching, it is binding, attaching. This is not moving here and there. This is just bending, detaching, bending, detaching, bending, detaching. Only the thin filament is moving. So from both the sides, the thin filament is moving, making the Z line to come closer. It shortens the length between the two Z lines. A band is remaining constant, same. And what about the H zone? It remains same. What about the I band? Shorten. This is what you will see in the MCQs. A band, I band, during muscle contraction, normal state, relaxed state. The relaxed state, normal state is the same, but confusing words. Okay, clear? So this is what is called as the muscle contraction. This is also called as excitation, contraction, coupling mechanism, or power stroke mechanism. Various determinations to explain the same concept. Okay, clear? This is how the skeletal muscle will undergo contraction. Clear? That's it with the muscle physiology.